first stage of protein synthesis is called transcription. This process occurs in the nucleus where the DNA is read to create mRNA, also the other RNA. The first step during transcription is called unwinding. This is where the RNA polymerase binds to a promoter at the start of the gene and unzips that section of DNA. The second step is called pairing. This is where the RNA strand is synthesized by complementary base pairing with the exposed DNA strand. The third step is called termination, where the RNA polymerase reaches the transcription terminator, which is a sequence of DNA that signals the end of a gene. Finally, we have processing, where the newly synthesized RNA molecule is released and processed. Now onto the second stage, translation. This stage involves ribosomes, rRNA, mRNA, tRNA, and amino acids. There are a few words we need to understand before we continue: codons and anticodons. Codons are mRNA that are read in 3 nucleotide segments, while anticodons are tRNA that are read in 3 nucleotide segments that pair with the codon on the mRNA. The first step, initiation, occurs when a small ribosomal subunit binds to the mRNA near the start codon. The first tRNA or anticodon will always bind to the methionine to begin the process, where later a large ribosomal subunit will join to bind, so other free-floating tRNA anticodons can bind to the mRNA codons. The second step is elongation, where the tRNA molecules and the mRNA binds. It adds amino acids to a growing polypeptide chain. This process begins in the A site, where the amino acid is being added by the tRNA. After, the protein slides to the P site, so the next tRNA can release its amino acid to form the polypeptide chain. After that, the ribosome is released from the E site. The third step, termination, happens when the A site of the ribosome reaches one of the 3 termination codons, UAA, UAG, or UGA, and the polypeptide is released. After the polypeptide is released, it will fold or twist into alpha helices or beta sheets due to H bonds. It will then fold even further due to functional group interactions. When multiple polypeptides come together, it can function as one protein, such as a hemoglobin. And that's all, folks.